Hello everyone, I'll be taking a practical topic determination of reticulocyte count. So, development, how the reticulocytes are developed. So, if you all know erythropoiesis, development of RBC, right? Then we know the development of reticulocytes, right? Because the RBCs develop after the reticulocyte, right? So, we'll see that it develops from pluripotent stem cells in the bone marrow. Then this undergoes the further differentiation and gives multipotent stem cells. Then it gives burst forming unit erythroid. Then it undergoes the differentiation and gives colony forming unit erythroid. Then it undergoes the further differentiation and gives pronormoblast. Now, as you all know, it develops from this actually the reticulocytes and RBCs are developed from the pluripotent stem cells, right? So as the cell gets matured, it decreases in the size. Okay. So as I said, colony forming unit erythroid, right? So the size of this cell is 19 to 23 micrometer, and now as it develops, the size will start getting decreasing. Okay. Then there are certain changes also takes place in a nucleus, in a cytoplasm, in appearance and presence and absence of hemoglobin, then in mitosis process. Okay, so it will be initially present and slowly it will start getting absent. Okay, so in case of colony forming unit erythroid, the size is on an average around 19 to 23, and now onwards the size will start getting decreased as the cell will develop. Right here, the nucleus is also large. Okay, and nucleoli is present on an average 4 to 5 in colony forming unit erythroid. Okay, hemoglobin is not present and mitosis is present. Now from there, proerythroblast develops. Now proerythroblast has nucleoli of 2 to 3. You can see the diagram of this proerythroblast or pronormoblast, anything we can say, right? Hemoglobin is still not present and mitosis is present. Now from here, early normoblast will be formed. So this is a picture of early normoblast. It is having a large nucleus, but nucleoli is not present. Here the nucleoli disappears and hemoglobin is still not present, but the process of synthesis of the hemoglobin has already started. Now mitosis is also present. Then from this, intermediate normoblast is formed. Here the hemoglobin appears. It is also known as polychromatic normoblast. Okay, this cell name is intermediate normoblast, or you can say polychromatic normoblast. Here hemoglobin appears and nucleus gets condensed. Okay, nucleoli is absent and mitosis is still present. Then from this cell, the late normoblast is formed. This is a picture of late normoblast. Now this has increased amount of hemoglobin. Okay, nucleoli is absent and mitosis is also absent. Now till intermediate normoblast, mitosis was present. Now in late normoblast, the mitosis is absent. From this cell, the reticulocyte is formed. The diameter of this cell is 7 to 7.5 micrometer. So as I said, the diameter of stem cell which was 19 to 23, and now the reticulocyte which is formed, diameter of the reticulocyte is 7 to 7.5 micrometer. So the size of cell gets decreased as it gets matured, right? Then in the reticulocyte, the nucleus is not present but still the ribosomal RNA is present, and due to which the hemoglobin synthesis still takes place. So this is a picture of reticulocyte. You can see. Okay. So this is a picture of reticulocyte. Now the reticulocyte remains in bone marrow for 1 to 2 days. Then it comes into the circulation, which remains in the circulation also for 1 to 2 days, and then it gets matured in a spleen. Okay. Now from the reticulocyte, the mature RBC is formed. Okay, so size of this is 6.9 to 7.4 micrometer. So the size of reticulocyte is little more as compared to the erythrocyte. Okay, erythrocyte is anucleated; it do not have nucleus. Okay, so now we'll see the next part, that is, what are the differentiating features of the reticulocyte from a erythrocyte. So as I said, the reticulocyte is a bit larger as compared to RBC. The size of reticulocyte is 7 to 7.5 micrometer, whereas size of RBC is 6.9 to 7.4 micrometer. And reticulocyte also contains a central core of RNA. Okay, and because of the presence of this RNA, when we are staining, it takes up a basic stain. Okay, so this line you will understand when I will teach you the further slides of practical. Now, what are the features of the reticulocyte? So reticulocytes are nothing but they are the juvenile RBC having a size of 7 to 7.5 micrometer and presence of ribosomal RNA, right? So normal count of reticulocyte we count in percentage, right? We count a total number of reticulocyte, then we convert it in a percentage. So here the values which are given in percentage, in newborns it is more, it is 30 to 50%, whereas in infant it is 2 to 6%, and in adult and children it is 0.2 to 2%. Okay. Now principle of this practical, that is reticulocyte count. So now what we do for this practical is we take a 1 to 2 drops of stain and we take 1 to 2 drops of blood, right? Then we mix this and then we make a smear from this mixture. Like how you make a smear in differential leukocyte count, the process of making smear is the same. You have to choose a spreader slide and then you have to place a slide, and from on that you have to make a smear. That is the same, right? So you take a stain, then you take a blood, you mix it, and then we make a smear. Okay, so this is the process. So now we'll see the principle first. So what this line says, the RNA content of reticulocytes, which is detected by exposing the living cells to the supravital stain. So what is supravital stain? What is vital stain? I'll be saying in the next slide. But imagine that one supravital stain is there. So now what we do, RNA content of the reticulocyte can be detected by exposing a living cell to the supravital stain. So that means we take a blood. So in the blood living cells will be present, and then we mix it with the supravital stain, and then we make a smear, and then we can identify the RNA content. Means that will get stained by this stain and we can identify the reticulocyte. So now these living blood cells which are mixed with the stain as opposed to differential leukocyte count where the smear is made before staining. This line says that the process of making smear, the method, that is the same, but the step is bit different as compared to differential leukocyte count, because in differential leukocyte count we were taking a blood, then immediately we were making a smear and then we used to stain that smear. But in reticulocyte count, stain and blood both are taken together and then they are mixed and then the smear is made. That is the difference. Okay. What stains are used for the reticulocyte count? Supravital stain and vital stain. Supravital stain. So what is supravital stain? So that is a dye which are used for staining the living cells in vitro. Dye's names I have placed in next slide. So imagine that dye. Okay, so that dye is used for staining the living cells in vitro. What is the meaning of in vitro? That is outside the body. So we use a dye to stain cells which are present in our blood drop which we have taken, right? So the dyes are used to stain the living cells in vitro. Means we take a sample of blood from our body and then we mix it with the dye. So that is supravital stain. But in vital stains, what we do is dyes are used for staining the living cells in vivo. Means we inject a dye inside our body, right? And then we take a sample. Okay. So that is the difference. Now, what are the content of stains? There are two stains which we are using, that is brilliant crescile blue stain and new methylene blue stain. Now in brilliant crescile blue, the picture of that is here. This is brilliant crescile blue. So content of this stain is brilliant crescile blue, sodium citrate and sodium chloride. The content of new methylene blue is, this is a picture of new methylene blue. So that is new methylene blue, sodium oxalate and sodium chloride. Here there is sodium citrate, whereas here there is sodium oxalate. Here there is brilliant crescile blue, here there is new methylene blue. Sodium chloride is in both. So once again, this is a picture of brilliant crescile blue, this is a picture of new methylene blue. Okay. Now what are the apparatus which you are going to use? That is instruments, right? So that is lancet to prick, right? Then brilliant crescile blue stain, then clean glass slide, okay, then blunt edge of glass slide to mix a stain and blood. As I said, you have to mix the stain and the blood, okay. So with blunt edge of glass slide, you have to mix the stain and the blood, okay. Then petri dish, okay. Then spreader slide to make a smear. Then test tube and microscope. Now as I said here, test tube, right? So in this practical, you can use test tube or you can use glass slide to mix a stain in a blood. As I said, you have to mix the stain in the blood. So if you have taken a glass slide, means you have taken a glass slide and on that you have placed a drop of stain and drop of blood. Then you can use the blunt edge of another glass slide to mix these two. If you have used test tube, then you can take 2 drops of stain and 2 drops of blood and then you can mix in the test tube. So there you don't need the glass slide. Okay. So anything you can use, test tube or glass slide. Okay. So in the video I have shown you the test tube. Okay. Then microscope. Now procedure. Clean the glass slides thoroughly. Okay. If you are using glass slide, you have to clean it. If you are using test tube, you have to clean the test tube. Now take a drop of stain on a glass slide. Okay. If you have taken a 1 drop of stain, then you have to take 1 drop of blood. And if you have taken 2 drops of stain, then you have to take 2 drops of blood. What I want to say is the amount of stain and blood should be the same. Then make a sterile finger prick and get a drop of blood and then mix it, because you have taken a slide and on that you have already placed a drop, right? On that you have to place a blood drop and then take another glass slide and with the blunt edge of this glass slide you have to mix the stain in a blood. Then place this glass slide which you on which you have mixed the blood and the stain, that glass slide you have to place in a petri dish, that is moist petri dish. How you make a moist petri dish? That is, by taking a petri dish and place a filter paper and then place a few drops of distilled water on filter paper so that it gets wet and it creates a moist environment, and in this you have to place slide which is mixed with the blood and stain. So this step you are doing even in platelet count. I already showed in the platelet count video. Okay, there you are placing new bar chamber, here we are placing a glass slide. Okay, now you have to select a spreader slide. Okay, and place the edge of this spreader slide, any one spreader slide you have to choose, okay, which has a smooth edge, right? How we were choosing in differential leukocyte count to make a smear spreader slide, we were choosing in same way you have to choose a spreader slide. Okay, so now place that spreader slide edge on the mixture. On the mixture means which the slide which we have placed on in petri dish. Okay, so the edge of this spreader you have to dip into this mixture. Okay, as soon as you dip, you have to take one more clean glass slide, and as soon as you dip, you take out. Okay, once the edge gets spreaded with that blood and stain as you placed inside that mixture, then you take it out and place on another clean glass slide and make a smear. Okay, so transfer the portion of the mixture which is sticking to the spreader slide to the another glass slide. Immediately make a thin smear. In this way you can do 2 to 3 times, making 2 to 3 smear. Okay, minimum 2 to 3 thin smear you can make, and then you have to allow the smear to get dry. Okay, then examine it under the 10x and then find out the thin portion of the smear where red cells are evenly distributed, and then you place a oil drop on that and then see in 100x, and you can identify the reticulocyte and red cells. Okay, now what are the precautions to be taken? So staining time should not be less than 10 minutes. Okay, staining time should not be less than 10 minutes. Mixing of blood with the stain to be done gently and thoroughly prior to making a smear. Stained platelet granules and leukocyte granules must not be mistaken for a reticulocyte. Okay, and precipitated stain might also be getting mistaken for the reticulum within the RBC. So that also you have to see carefully. Now we'll see the video. So 2 to 3 drops of dye he has taken in test tube. Then 2 to 3 drops of blood he has placed in test tube. Right? Here, see here it is written that if the RBC count is too low, add more volume of blood, and if it is too high, then add less volume. What do you understand by this sentence? Because as I said, in this practical, you have to take equal amount of stain and dye. Stain and blood. But if the patient's hemoglobin is less, RBC count is less, if the patient is having anemia, then what you have to do? You have to take more blood, right? That time the amount of blood is slightly more than the stain. And if the patient is having more HB and more RBC count, okay, means the patient is having polycythemia picture, then the amount of blood should be less as compared to the amount of dye. Okay, so that you have to remember. Then here they have kept this mixture. As I said, you have to mix the stain in the blood, right? If you have taken glass slide, then glass slide. If you have taken test tube, then test tube, whichever you have taken, right? But you have to mix it. Then after mixture, here they have keep the mixture at 37 degree for 15 to 20 minutes, right? This is incubator. They have placed in incubator. If in laboratory incubators are available, you can place it in that. Otherwise, also you can, instead of this also, which step I said, what you do in platelet count. In moist petri dish, you can place a slide, right? There you were in platelet count, you were placing a newborn chamber. Here in reticulocyte, we take a petri dish in that wet filter paper and then we place a slide. So this step and that step is a similar. Okay. Now take the from this incubator and then you have to make a smear. This is what is made a smear. Then you have to see in the microscope once the smear gets dried. Okay. And you have to choose an area where the cells are evenly distributed. Okay, and staining is good. And once you get a good area, there you have to put the oil and see under 100x. Now, how the reticulocyte and RBC look. So this is the picture of reticulocyte and RBC. So these are reticulocytes, okay, and these are RBCs. So reticulocytes are fine, deep, violet filament with granules arranged in a network. See, you can see. And RBCs are slightly smaller in size than the reticulocytes. So these are RBC, these are reticulocytes. Okay. Then you have to count the reticulocyte and RBC in each field. Okay, minimum of 1000 cells you have to count with the minimum of 10 fields. Okay, this is a picture of RBCs and this is reticulocyte. So now you can make a table like this while counting that 10 fields. Then in first field, for example, we are seeing first field. Think that this picture is of one field. Okay, so you have to count. For example, this is first field. How many reticulocyte can I see? 1, 2, 3. Then you have to write here 3. And how many RBCs? Then you have to count RBCs. Then you have to write here. Okay, and then total number of cells. So this is how you have to see 10 fields and you have to count a number of reticulocyte, RBC and total cell. Now how will you count? Once you complete counting reticulocyte, now final count. That is number of reticulocytes which are counted. See here, whatever number of this, it comes total number. Okay, so that divided by total number of cells which are counted. That is this total number of cells. That is reticulocyte plus RBC. So this divided by this into 100. Then you get reticulocyte in percentage. Okay, for example, 15 reticulocytes I could count. So 15 divided by total cell which I counted was 1500 into 100. So that comes 1%. Now, as I said, supravital stain you can use for reticulocyte count. Okay, but it also stains. It do not stain only the reticulocyte. Okay, it also stains Howell-Jolly bodies, Haynes bodies, Pappenheimer bodies. These are the inclusion bodies which are present in different pathological conditions. This Howell-Jolly bodies is dark blue to purple color inclusions. These are seen in splenectomy patient or in case of pernicious anemia patient, hemolytic anemia patient. This Howell-Jolly body is present. Haynes body is present in glucose 6-phosphate dehydrogenase deficiency. Then Pappenheimer bodies. So these are present in iron-loaded anemia, and it is also seen in case of the hyposplenism. Okay, so these are the bodies which are seen in different pathological condition. Okay, so that you have to differentiate from when it is present. You have to count it separately. Okay, this should not be included in reticulocyte count. Okay, then What are the indications when you should do the reticulocyte count? So that is to follow up therapeutic response of a patient having anemia. Means when patient comes to us and if we find their hemoglobin is less, when you do RBC count, based on that when you come to know that the patient is having anemia, okay, what we do, we give them a therapy, right? We give them a drug. Then when they come for the follow up, we come to know, we should identify whether they are responding well or not to our drug therapy, right? So based, that is to follow up therapeutic response of anemia. Means so that time, when we are giving a drug, means on response to this, bone marrow will start getting good activity and it will start producing cell. Okay, so based on that we can come to know, so bone marrow activity will become good and the reticulocyte will also increase. So we can come to know that the patient is giving response to the therapeutic treatment. Okay, then in hemolytic anemia, okay, also we can do the reticulocyte count, there it will decrease. Okay, because there will be the destruction of RBC, so the count will be less. To know the activity of bone marrow, see if the count of reticulocyte is more, means the bone marrow is active more. If the count of reticulocyte is too less, then the bone marrow activity is less. Based on that we can come to. Now causes of reticulocytosis and causes of reticulocytopenia. Okay, reticulocytosis means increase in reticulocyte count and reticulocytopenia means it is decreased in reticulocyte count. Okay, now reticulocyte count is increased in case of physiological conditions are newborns, infants, in case of high altitude, right? When the a person goes to high altitude, what happens is their cell count will start getting decreased because of there is okay. So what happens, as a compensation the bone marrow will start producing more and more cells. Okay, moderate to severe exercise. Okay, so these are the physiological cause. Now in case of hemolytic anemia where destruction of cell takes place, okay, so as compensation bone marrow will produce more. Okay, so in case of acute hemorrhage there also blood loss is there, so cell count will decrease, so there also as a compensation bone marrow will produce more cells. During treatment of deficiency anemia, which I already said, any condition that stimulates the bone marrow to produce more RBC, there also more reticulocyte is there, and heavy smoking. So these are the causes. Then reticulocytopenia causes aplastic anemia where the activity of bone marrow is not good. Okay, it is suppressed. In case of myxedema, hypopituitarism, leukoerythroplastic anemia, in all these conditions, the reticulocyte count will decrease. Thank you.